Good afternoon, everybody. We'll get started. I know a large part of our audience is online, so hopefully everybody can see us and hear us. So it's my honor today to introduce our CTSI distinguished speaker, Dr. Ken Poss. I've known Ken for many years and personally followed his work over the last 1.5 decades. And it seems a long time, but it seems only yesterday that when, as a postdoc, we were made aware of this star junior faculty who had come to the cell biology department at Duke, who had published this amazing but very simple paper on heart regeneration in zebrafish. And that paper and Ken's subsequent work really ushered in a new field of heart regeneration and has led, as everybody knows in this room, to really hundreds of papers across many, many labs in this country and across the world. And more than that, it actually inspired new investigators to try out the mammalian model of heart regeneration in the neonatal mouse, the rat, and now even in larger animal models. And it's all really a legacy and a testimony of Ken's original work in the zebrafish heart, which led to this sort of exponential growth in the field of heart regeneration. Ken has also recently ventured into looking at appendage regeneration, retina and spinal cord regeneration, and continues to immensely contribute to this field. His background, he's completed his PhD with the Dr. Tonegawa at MIT and then postdoc with Mark Keating, initially at Utah and then at the Children's Hospital in Boston, where he did his work on zebrafish regeneration. He was then recruited to the cell biology department at Duke, where he has remained most of his career. Enormously successful. He was director of Duke's Regenerative X initiative, and then headed the Institute of Regeneration at Duke. He's recently moved to University of Wisconsin-Madison, where he directs the Regenerative Biology Institute at the Mogridge Institute of Research. Ken has also enormously contributed to the sort of growth of the field. He's been founding president of the International Society for Regenerative Biology. And not just as a superb scientist, Ken is also a beloved mentor, having mentored more than 15 of his postdoctoral and trainees and graduate trainees have academic tenure track positions at leading US universities. So it's always a treat to listen to Ken and update his, have him update his story on heart regeneration. So Ken, welcome to UCLA.
Good afternoon, everyone. Can you hear me okay? First of all, a big thanks to Arjun for a very generous introduction. I don't know if I'm beloved by my former postdocs, but I'd like to think so. I mean, I will for this moment. It's great to be here at UCLA. It's been, we were saying about 10 years since I was here last. So exciting to come again. And I thank everyone who I've met with already and who I will meet with for them spending their time with me. My group, as Arjun mentioned, is very broadly interested in how and why tissue regeneration happens. And for many years, we've been helping to push the value of the zebrafish model for getting at those questions of how and why. And we study many examples of regeneration in zebrafish. Some of these are highly relevant to regenerative medicine for the human condition, for instance, as I'll focus on today, and as generously alluded to, zebrafish are probably the best at regenerating heart that we know of, and maybe we know the most about how it works after all these years. They can also regenerate after a full transection of the spinal cord, a paralyzing injury. Within several weeks, they're swimming like nothing happened. We've begun to study how they regenerate the kidney. These animals can grow new nephrons throughout life, and they can regenerate after massive loss of nephrons. And then finally, on these bottom videos, we're interested in examples where you just see the basic beauty of regeneration. For instance, how animals like teleosts can regenerate their fins after complex amputation. Or on the right here, this is a plucked scale with labeled osteoblast that's regenerating that we can use live imaging to capture videos of. The focus of the talk today is mainly the heart, and as I think all of you know, this is not one of our best tissues at regeneration. With a very basic review, when someone has a heart attack, as you know, there's an immediate loss of up to, in some cases, 1 billion heart muscle cells. Now, 90% of people will survive a heart attack, but the response to this is not regeneration of new heart muscle, but it's efficient formation of a scar. And many people do quite well with the scarred hearts, but it can lead to complications. It can lead to a second heart attack and eventual heart failure. So I think most cardiovascular researchers, most cardiothoracic surgeons and cardiologists would agree that it would be a good thing to find some method or methods to regenerate healthy new heart muscle rather than a scar. So this has been a focus of regenerative medicine for many years. Now, there are many phenomenal groups in the field of heart regeneration using different approaches, for instance, to generate mature cardiomyocytes to inject into an infarct, or maybe a patch of cardiomyocytes, or reprogramming of some sorts. But as a developmental biologist, as I grew up as, we really look for a role model, something that does this process well and can teach us how it may be applied, say, in the human context. And at the time, when I was a postdoc with Mark Keating in the early 2000s, there just wasn't a role model that could tell us how heart regeneration happens. And really, there wasn't really a large group of people who believed that heart regeneration was a thing. Now, there have been groups that had worked on it, and I won't go through all the ancient literature, but many studies in the 50s, 60s, and 70s, well, I won't say many studies, but a handful of studies, some of these from Soviet groups, looked at the possibility of a regenerative response in, say, a leopard frog or salamanders like a newt or the axolotl here. And the data suggested there was probably something interesting going on. It looked like in electron micrographs that cardiomyocyte nuclei were spreading their chromosomes, and maybe those cells were undergoing division. But the actual tissue response to the injury was kind of equivocal. Some reported scarring, some reported possibly regeneration. And so toward the end of my postdoc, we reported what was a simple paper, and it was just, I think, unequivocally, if you cut off a quarter of the ventricle of an adult zebrafish heart, the scarring response will be minimal, sometimes absent, and the dominant response is the growth of new heart muscle. And we reported some of the basic mechanisms in this story, but what we were most excited about is that zebrafish were this up-and-coming genetic model, and that we could eventually and even start at the time using molecular genetics to approach this phenomenon of innate heart regeneration. And so that's shown here very basic histology from the old days. But if you remove 20% of the ventricle, cut right through the lumen, you get some massive bleeding, but clotting occurs quickly within a week, a nice vibrant clot. And then within a month or two, you don't see replacement of that tissue by a scar, but you see new muscle formed at the injury site. Now, there have been many, many groups who have entered. Oh, something happened. Should I press OK? Yeah, I think so. And go back to slides. How do I do that? Here? No.
Yeah, 1 second.
Okay. I was saying there's no. I think there's probably a slide here that's messing this up, so I will probably skip a slide. I think I'm going to skip ahead a couple of slides.
This happens again. There might be something, an issue with the PC. Might have to go towards the laptop.
Yeah, it's pausing at that slide. So,
Right,
So,
this doesn't look like
can
that
you
slide.
if you set it up ahead of that slide?
Ahead of this, no problem.
Yeah, let me.
Good.
I'm gonna go right here. Okay, that was the one.
Right.
To here. Safe mode.
No.
This is also a video, so.
Let's see what happens. Okay, good. So far. So
Okay,
good. Let's try that again.
That was an important summary slide of the whole field of heart regeneration. And it truly was. It was embarrassing that maybe I tried to put too much into that slide. But you know what I was going to say is that first, if you're to remember one thing about how heart regeneration works in its innate form, it's not a stem cell based mechanism, but in zebrafish and other fish and other amphibians, and then even in neonatal mammalian contexts, like Arjun referred to, developed by Hesham Sadek and Eric Olson's lab a long time back. it's clear that cardiomyocytes are the source for that new muscle that comes back. And that's what one thing you should take, because I won't go extensively into all the mechanisms of heart regeneration that we know about. I think the other component that is a critical aspect of the field is first,